Shortness of breath, wheezing, and in the worst cases, even death. Asthma. In Britain alone, it affects 1.5 million children. Yet the disease was hardly known a century ago and troubles just the rich nations. It's still rare in the developing world. Just why remains a mystery, as elusive as a cure. But a British scientist, Dr. John Stanford, believes that in the African soil lies an answer, not only for asthma, but for a host of other diseases as well. John Stanford has found a microorganism that seems to unlock the incredible power of the body's ability to fight illness. If he's right, it's a bacteria that will change medicine.
This organism corrects the immune system in a beneficial way, and we're hoping that from it we can produce reagents which are going to be of value in a whole series of chronic diseases.
Germs have a bad reputation.
When handling food, avoid coughing on it or sneezing on it or breathing on it.
For centuries, we've been taught to think of them as the cause of disease.
And don't, repeat, don't ever do this sort of thing. Use your mouth and breath for singing or whistling or playing a mouth organ, but never, repeat, never, for blowing open a paper bag into which food will be put.
Modern culture tends to equate all germs with devastating disease, while in fact the vast majority of microorganisms are harmless to us, and some absolutely essential for our well-being. Our obsession with destroying bugs may have gone too far. Professor John Stanford and immunologist Graham Rook are among a growing number of scientists who believe that too little contact with bacteria may be bad for our health, and it may actually cause disease.
It's entirely possible that the increasing incidence of disorders like allergies and autoimmune diseases is due to man's increasing isolation from bacteria in the environment.
Just like the brain, the immune system needs to learn. And part of the learning process that the immune system goes through is in fact via meeting germs, bacteria.
If the immune system doesn't have this educational input and doesn't get properly trained, it starts looking for a fight and gets out of control.
If you haven't met sufficient germs in the early development phase of your immune system, then you overreact when you meet allergens. And the result of this are the allergic diseases.
The idea that contact with bacteria is essential to our health is a theory that recently gained general acceptance in the scientific world. But for 30 years, it's been the everyday observation for Stanford. He studied man's relationship with bacteria at the front line of medicine, travelling the developing world, dealing with diseases of the poor, like leprosy and tuberculosis. With him, he usually took his entire family, including his wife and collaborator, Cynthia. They travelled where few would venture: Uganda, Latin America, Nepal, even to a leprosarium in war-ravaged Iran.
We rolled up with the family. The children helped. Even 10-year-olds can weigh and measure. I think also because we weren't a perfect family, because Matty was retarded, because Mark had his problems as well, because we had an adopted child among us. And I think if just John had gone by himself, he wouldn't have been welcome at all. He would have been seen as using people.
With his background in bacteriology, Stanford was the right man in the right place to make a crucial observation. that an otherwise innocuous bacteria called M. vaccae might make a dramatic contribution to treating an extraordinary range of diseases. From a mud puddle in Africa, he began a scientific odyssey around the world that is about to reach its climax.
The story behind Mycobacterium vaccae is some lovely detective work by my colleague John Stanford, because he noticed that the incidence of leprosy and of tuberculosis, the two major diseases caused by the pathogenic members of this group of bacteria, that he showed that the incidence of these two diseases was different in different parts of the same country and appeared to correlate with the geography of the countryside in which they were living.
The whole concept of the importance of the contact between man and the environment struck me first when I was working out with my family in Uganda, when we were particularly interested in the distribution of tuberculosis and leprosy and why BCG had been so successful there in the prevention of leprosy.
It has been known for some time that BCG, the vaccine usually used to prevent tuberculosis, was sometimes also effective against leprosy. Many trials were conducted in the 1970s to test its effectiveness, and for some unknown reason, the best results were found in one isolated region of Uganda, Lake Kyoga.
We were particularly keen to find out, since the effect on the vaccine was not worldwide and that BCG doesn't work everywhere, it only works in particular areas as well as it did in Uganda, and we wanted to know what was it that was special about the environment there that allowed that to happen. And it was from the bacteriological studies of the soil samples that we identified a whole series of different mycobacteria, most of them nothing apparently to do with disease, and amongst them was an organism called Mycobacterium vaccae.
Of course, it turned out to be an organism which shares many of its proteins with these pathogenic organisms, leprosy and tuberculosis. The immune system sees proteins of Mycobacterium vaccae as though they were in fact proteins of leprosy or tuberculosis. The proteins are extremely similar in the two groups of organisms.
Though very similar, one bacteria can kill, while the other can prevent disease. And the richest source of helpful mycobacteria was along the shores of Lake Kyoga, where people came to bathe and cook every day.
And when we put this together with the distribution of tuberculosis, leprosy and the efficacy of BCG vaccine, we realised that what determines man's susceptibility to disease or ability to respond to a vaccine is in fact the environment and what he meets in that environment.
It was a radical theory at the time, and to test it, Stanford conducted an experiment back in London at the Middlesex Hospital. M. vaccae was cultured from the Ugandan mud samples and added to the BCG vaccine. The tests indicated that animals given M. vaccae with their BCG were much better protected against leprosy than the ones that only received BCG. It was an important first step that others would later confirm in more detail. M. vaccae seemed to be doing something highly beneficial to the immune system.
It's now generally accepted that organisms related to leprosy and related to tuberculosis, like Mycobacterium vaccae from the environment, can actually vaccinate people spontaneously. So people living in the appropriate environments and meeting these organisms are to some extent protected from these diseases anyway.
Stanford's vision that the health of our immune system depends on our daily contact with environmental microbes had taken a crucial step forward once he tested M. vaccae in the lab. But after years of seeing firsthand the suffering of so many people from disease, Professor Stanford's true aim was to see if he could put his research to medical use. M. vaccae had never been known to cause any disease, and he decided to try it first in the prevention of mankind's most stigmatized illness, leprosy. But to be sure it was safe and had no side effects, he had to test it first on humans, and that meant injecting the bacteria into himself and his wife Cynthia.
John and I injected each other to begin with because on the principle that you can't give to someone else if you're not prepared to have it yourself.
The first ones I had were back in 1975. and I've had them on and off ever since. When I used to make the material before every batch, I used to test on myself, because I never wanted to give people an injection which would produce a severe response.
In July 1978, the whole family went to Iran to evaluate the use of Mvaki in the prevention of illness in the children of leprosy patients. They lived in Baba Bari, in a leper colony near Tabriz. Leprosy can lie dormant for many years, and it would take a decade to see if the trial had succeeded.
It was when we went back to Baba Bari at the height of the Iran-Iraq war, when we had an invitation to go back there, and all the children that we'd vaccinated 12 years before, they were straight, they were tall, they were beautiful, and there wasn't a hint of leprosy or a case of tuberculosis in all that time. And then I knew it really, really worked.
Now that Mvaki had shown itself to be successful in the prevention of leprosy, the next step was to see if it could help those who already had the disease. And that took Stanford and his family to a leprosy hospital in Fontille, Spain.
It was from this laboratory in Fontilius that some of the early and very important work was carried out on Mycobacterium vaccae at a time when we were moving in concept from a prophylactic or preventive to a therapeutic or treatment.
Like so many leper colonies, Fontille suffered from people's unfounded fear of the disease. It was separated from the outside world by a stone wall, even though its patients presented little danger. The infectious stage of leprosy can be cured with antibiotics, but for the patients, the problem continues. Their immune system keeps attacking the body long after the last leprosy bacilli has been defeated. What started as an infection has become an autoimmune disease. Stanford and his Spanish colleague, Pedro Torres, were interested in seeing if Mvaki could help.
When we first came here, we started off with the dose which we'd determined for preventing disease.
And continue to increase yearly.
And then we went up tenfold to a 10 times higher concentration, and we observed those patients for a year, and then we went up tenfold more.
Until we reached what we thought was the optimal dose for treatment.
And at this point, we got the breakthrough in what we were looking for in the immunity, and we knew that we defined at least the beginnings of the therapeutic range for treatment.
Mvaki was able to overcome the massive imbalance in the leprosy patient's immune system. The Spanish experiment established that Mvaki was not just a preventative like a vaccine, it showed clearly that it had great potential as a treatment for established disease. If Stanford was right, his discovery could make its presence felt in other diseases too.
One of the first of these to turn up was observed by one of my colleagues, Dr. Ramu, in southern India, who found that amongst the leprosy patients that he was treating with Mvaki was one with, coincidence, psoriasis. And he went on to take a group of 9 or 10 cases of psoriasis without leprosy and show that the immunotherapy was also effective in them. And once this had been picked up, although we've been saying there must be other diseases that will affect, this was the first one that was really clearly recognised.
In fact, Cynthia Stanford, who had a vascular disease, may have been the first to benefit from the vaccine.
I had it, and after the little pink bump had gone down, I sort of forgot all about it until we got through to the winter, and I suddenly realised I was at Christmas, and the Raynaud's disease that I'd had ever since I was a child wasn't there anymore. Well, this was unheard of, because before that, quite often I couldn't find the money in my purse. My fingers were so clumsy, handling keys was difficult. In our family, there are lots of people who suffer from Raynaud's disease, and since the cure was at hand, they all lined up to have it. One was my mother, who had cancer, including spinal secondaries. One was my sister, who had arthritis, and another daughter had arthritis as well. One had asthma, one had schizophrenia. Anyway, they all lined up for the injection for their Raynaud's disease. And then I noticed that the other things had disappeared too. Mother's cancer regressed and her spinal secondaries disappeared. Thomasina's asthma disappeared for 3 months.
I just don't need to take my inhaler so much before any time I did any sort of physical activity, you know, from a child running around the playground right through to an adult and doing any sort of physical work in the garden or anything like that. I'd need my inhaler a lot, but now I don't need it nearly so much, maybe 2-3 times a day as opposed to 14-15 times.
The family observations were intriguing, but still required scientific investigation. What is clear, though, is that right now we need a breakthrough. While medicine has made tremendous advances, in the new century we face a new challenge. Our sterile society has a health crisis. New illnesses, allergic diseases in particular, are skyrocketing. Asthma alone afflicts between 20 and 30% of people in countries like Britain and Australia. Autoimmune diseases like diabetes and multiple sclerosis are also on the rise.
The unifying thing about all three diseases is that they are all disorders of the regulation of the immune system.
What's changing in the environment of developed countries that is leading to an increase in these diseases? The evidence suggests a surprising cause.
One of the first things to be noticed was that you were less likely to be allergic if you came from a large family with lots of siblings. It particularly helped if you have older brothers. They're better than older sisters, which was interesting because little brothers are probably dirtier.
A study involving thousands of children showed that allergic reactions were influenced by how often the children washed their hands and faces. Those that washed less actually had better protection against allergic disease.
Further work has been to look at children brought up on farms, and this seems to be true whether you look in Scandinavia or in Germany or in Switzerland, that children that are brought up on farms are less likely to have allergic symptoms than exactly equivalent children from the same sort of economic background living in the same villages but not actually brought up on a farm. All of these things, therefore, tend to suggest that organisms in the environment might actually be good for you in terms of protecting you from allergy.
It's a hypothesis that begins to solve the mysterious rise of allergic disease, and also points to an explanation as to why a harmless bacteria like M. vaccae could yield remarkable results.
Man evolved to live close to his environment. In fact, his immune system became dependent on that environment for one of its most important early lessons, the lesson which would result in our being able to overcome the problems of allergy and of autoimmunity and of other diseases for the rest of our long lives.
One problem is that we're born with a large repertoire of white blood cells which recognize not only foreign proteins in bacteria and so forth, but also our own proteins. Those cells have now got to be educated. You've got to have a mechanism that comes in and says to those cells, which can recognize you or the bacterium, that they should attack when it's a bacterium, they should not attack when it's you. You're born with the hardware, but you need a lot of software and information put in after birth for them to function correctly.
During the progress of Western civilization, this vital contact with harmless bacteria is lost to many of us. We have moved from countryside to cities, lost contact with animals, and especially the soil.
You see this only too easily in looking here at the terraces. The terraces were made, and at one time there were thousands of people working on them. Nearly everyone went out from the very earliest times into the fields, and they lived in houses with earth floors. Today all of that has changed. At first people thought that the more we were separated from germs, the better it was for us. But now we begin to realise that this is not right. any stone you pick up will have germs living under it. Any bit of dust will have bacteria living within it. And amongst these bacteria are a group called the mycobacteria. And these are particularly important because they contain in their surroundings, their cell wall that wraps around them, substances which drive the immune system in very important ways.
We think that they may be the ones that, because they were there throughout mammalian evolution, are now an actual necessity to the immune system. The immune system expects them to be there, it's evolved with them there, they have to be there, but they're not there in a concrete environment or in treated water, chlorinated water, such as we now all drink. What we do need to do is find out what we have lost through modern hygiene and put it back. Contaminating the environment with organisms deliberately, which are protective, is certainly an option. Why shouldn't we spray them into the environment? Why shouldn't we spray them into the house dust? There are many possibilities. We may also want to make new vaccines which actually replace that learning input that is no longer present in our environments.
Which is exactly what Stanford and Rook are doing. With the backing of the University College of London, they have formed a public company to develop new treatments based on MVACI. It is an expensive and complex process that involves working with many other independent scientists in clinical trials. The vaccine for the trials contain harmless MVACI, cultured from the original African mud samples in the specialist laboratories at Camar, Porton Down. It can then be used in closely controlled human trials, among them those of asthma specialist Dr. Rathko Djukanovic at Southampton General Hospital. He is looking for a way to stabilise the overreacting immune system of his asthma patients.
Those of us interested in allergic diseases such as asthma have for a long time been looking for a magic bullet which would give us the possibility to redress the balance.
The apparent ability of MVACI to act as a switch on the immune system convinced Djukanovic to put MVACI to the test.
Dr. Djukanovic.
Hello.
Nice to meet you. What we're trying to see is the effect of a vaccine that has been found to have a potent regulatory effect on the immune system.
The results of the small trial were very encouraging. After only one injection of MVACI, asthma symptoms were reduced on average by 30%, and in 2/3 of the patients by even more. Mark Dyer was one of them.
Big Hablo, keep going, wonderful, that'll do lovely.
I don't have to think anymore of where's my medication, where's my Ventolin. I used to take it around with me everywhere I went. And there's never any more nagging doubts in the back of my mind as to have I got enough Ventolin, am I going to suffer from an asthma attack today?
Although it's early days, nevertheless I think the results of the trial are very exciting. What we have now is proof of concept, which we have to take further.
MVACI has shown itself to be effective in dealing with entirely different diseases, like leprosy and asthma. But its powers may not stop there. It might have a role to play in beating the greatest medical challenge of them all: cancer. In 1988, British professor John Grange observed that the BCG vaccine was beneficial in treating the disease. MVACI may do even better.
Curiously, cancer patients' immune systems, although they don't develop allergies, nevertheless they have a shift in the balance of the immune system in rather the same direction as the shift that occurs in allergic people. So by Mycobacterium vaccae helps to turn off that part of the system which is not good for dealing with cancer and switch on the part of the immune system which is good for dealing with cancer. And then the immune system just does the job all by itself.
A number of cancer trials are underway, and Dr. Mary O'Brien at the Royal Marsden Hospital, London, leads one of them.
For decades, doctors have tried to use the immune system as a treatment of cancer, in a way trying to trigger the immune system to recognize something that is foreign in their body, i.e. a cancer. Around the beginning of 1996, I was very impressed with the results that I saw in laboratory models of cancer using MVACI At that point, I tried to think how we could apply this to patients with cancer and in my own practice, patients with lung cancer. Good morning, Mr. Morris. Now then, what sort of night did you have?
Not too bad.
Good. You all ready to have this vaccine?
Yes, Doctor.
Okay. If you just lie down there.
Initially, Dr. O'Brien used MVACI in combination with chemotherapy on a small group of last-stage lung cancer patients.
To our surprise, we find that patients receiving MVACI plus chemotherapy did better than we expected. And even though that began in 1996, we still have some survivors from that study, and they were all treated with the combination. In medicine, we then proceed to confirm our results because many things that we see in small pilot studies are actually due to selection of cases and a little bit of luck on the part of both patients and investigators. To confirm that this is a real scientific observation, we have to do a large study, a randomized study, what we call a phase 3 study.
This larger, so-called Phase 3 trial has now studied 400 lung cancer patients from around Europe to test the effectiveness of combined MVACI and chemotherapy.
If the phase 3 study is positive, showing that the addition of MVACI to standard chemotherapy improves survival by a small amount, but real, reaching statistically significance, then I think we're onto something that we haven't had in medicine for a number of years.
30 years after it began, John Stanford's scientific journey has reached its final stage. The promising clinical trials now need to be replicated widely, and then official approval lies ahead.
It looks incredible now to look back that the simple observations that were made in a car coming down through Uganda have played out so well. But in fact, when you look back on it, you can think, as so many of these things that are really very simple, that had you just laid in the bath for long enough, you would have been able to turn this up without actually having to go anywhere.
Once you've been working towards something for a very long time, when you arrive at it, and when one can see the full thing unfolding in front of you, it's lovely.
It's the first time that something has been taken from the environment and been used to modify the immune system in a way that will influence a whole lot of diseases.
At present, we regard it as a potential treatment. That doesn't mean that in future, with increased knowledge of how to use it, improved regimens, getting exactly the optimal doses and frequencies, then maybe we can go beyond mere treatment. But at present, we certainly can't say that.
At first, of course, it's going to have to be aimed at developed world diseases. Perhaps cancer will be the first, or perhaps allergies will be there. But there's no doubt at all that as soon as we can, it's very necessary that it's used for the developing world, which is where it was born and what it was originally designed to do.
One of the side products of our research is the charity. We try to help the people that are disabled by both poverty and disease. It's simply not on, I don't think, to make discoveries in the developing part of the world and then exploit them in the developed part of the world. One has to redress the balance.